Welcome back to Acing AP Biology. Today, we will be diving deep into organelles and their special functions in the cell. Let's begin with the endomembrane system. Recall that eukaryotic cells have membrane-bound organelles, which divide the cell into different compartments. The endomembrane system includes the nuclear envelope, the endoplasmic reticulum, the Golgi apparatus, vacuoles, and lysosomes. Interestingly, the plasma membrane is not a part of the endomembrane system, but it is related to it. Membranes may interact with each other directly or through vesicles, which pinch off the membrane and then move to another location in the cell. Membranes are dynamic, meaning that they are constantly changing in structure, size, and activity. The nucleus is the largest organelle in the cell. It is known as the control center because it contains the genetic material that codes for cellular activity. The nuclear envelope is the double membrane surrounding the nucleus. It contains nuclear pores that let materials in and out of the nucleus. Genes are organized as chromatin, which are composed of DNA and histone protein. When chromatin is condensed, it becomes chromosomes. Human body cells have 23 pairs of chromosomes. The nucleolus produces ribosomes. The nuclear lamina provides support, shape, and transportation. The protein manufacturing process begins in the nucleus. DNA is transcribed into mRNA. Then, the mRNA moves through the nuclear pores into the cytoplasm and attaches itself to a ribosome. Ribosomes are protein manufacturers. They are composed of a small subunit and a large subunit, made up of rRNA or ribosomal RNA and protein. mRNA coming from the nucleus attaches to the ribosome and the translation process begins, where the mRNA code is translated into an amino acid sequence, which is the primary structure of proteins. Ribosomes can be free in the cytoplasm or bound to the endoplasmic reticulum. Proteins produced by free ribosomes usually have functions within the cell. Proteins produced by bound ribosomes most likely have functions either in the plasma membrane or outside the cell. In general, the endoplasmic reticulum, or the ER, is the most extensive component of the endomembrane system. It consists of tubules and cisternae, or sacs, with interior spaces called lumen, which are separated from the cytosol. The ER is continuous with the nuclear envelope. The smooth endoplasmic reticulum does not have ribosomes. It produces lipids such as phospholipids and steroids. It also detoxifies drugs and poisons. In fact, alcoholics have a lot of smooth ER in their cells. The rough endoplasmic reticulum is deemed rough because it is embedded with ribosomes. The rough ER is involved with producing secretory proteins as well as plasma membrane proteins. Once the mRNA attaches to the ribosome, embedded in the rough ER, the translation process begins. The new polypeptide chain is then threaded into the lumen, or the internal space. Here, the protein folds into its native conformation. Sometimes a carbohydrate is added if necessary for that protein's function. Then the protein moves to the transitional end of the ER and pinches off in a transport vesicle. The protein manufacturing process now continues in the Golgi apparatus. The Golgi apparatus receives protein from the rough ER and modifies, sorts, and labels it. The protein comes in from the cis phase of the Golgi apparatus as the transport vesicle from the ER merges into the cis phase of the Golgi apparatus, and then the protein is secreted into the cisternal space. Once the modifying process is complete, the protein exits the Golgi via transport vesicle and moves to other sites. If the protein is destined to leave the cell, it will go to the plasma membrane and be secreted out. This process is called exocytosis, which we will talk about in detail in the next unit. To end this video, I wanted to summarize the protein manufacturing process because this is often tested on the AP. It begins with DNA in the nucleus, which is then transcribed into mRNA. Then the mRNA moves out of the nucleus and attaches itself to a ribosome. If this protein is destined to be a secretory protein or a plasma membrane protein, the ribosome embeds itself into the rough ER, and then begins the translation process from mRNA to an amino acid sequence. The growing polypeptide chain is inserted into the lumen of the rough ER, where it folds into its unique native conformation. It exits the rough ER in a transport vesicle moving to the Golgi apparatus, where it is further modified. It leaves the Golgi apparatus through a transport vesicle and goes to the plasma membrane, where the protein is secreted. In the next video, we will continue discussing the specialized functions of organelles and cell-to-cell communication. See you then. For more bio content, hit subscribe.